How to use genetic biocontrol to manage insect pests. Pest insects often transmit disease to humans and animals and destroy crops. Controlling these pests can improve human health and increase agricultural production. Pest insects are often controlled by releasing other insects into the environment that will specifically kill the pests. This method of pest control is called biological control and is environmentally friendly because it reduces reliance on chemical insecticides and targets only the pest insects. Genetic biocontrol is a type of biological control where the released insects are the same species as the pest, but with critical genetic differences and traits that affect their reproduction or lifespan. The released insects transmit those genetic traits to the pest insects in the field, leading to sterility or premature death of the pests and a reduction in the pest population. Genetic biocontrol is used widely throughout the world to control many types of insect pests. Africa has a long history of using biological control and genetic biocontrol. One of the earliest demonstrations of genetic biocontrol involved the local elimination of tsetse flies from an area in Tanzania 80 years ago. More recently, sterile male tsetse flies were used to successfully eliminate the insects from Zanzibar and parts of Senegal. The release of sterile male insects is a genetic biocontrol method known as the sterile insect technique. In South Africa, millions of dollars in fruit production are being protected from multiple devastating insect pests using the sterile insect technique. A devastating invasive insect pest of cattle called the screwworm was eliminated from tens of thousands of square kilometers of Libya using the sterile insect technique. Although there has been much success using genetic biocontrol, most current technologies, such as the sterile insect technique, require regular, continuous release of the insects with the unique genetic traits to achieve sustained control of the insect pest populations. Advances in genetics and biotechnology are expanding the possible applications of genetic biocontrol, providing more sustainable methods to control insect pests. The first experimental release of a genetically modified mosquito in Africa occurred in Burkina Faso in 2018 as part of a mosquito genetic biocontrol development effort. Successful efforts to create genetically modified mosquitoes are also occurring in Tanzania. These new biotechnology-based approaches enable genetic traits carried by the released insects to rapidly spread or drive through the target pest population by a process called gene drive. Gene drive genetic biocontrol has the potential to be sustainable and effective over large remote areas, leading to the reduction or genetic modification of target pest insect populations. Gene drive is different from other genetic biocontrol techniques, such as the sterile insect, in that the genetic traits are passed to the field pests from generation to generation, thus reducing the need for continuous release. If used against human malaria-transmitting mosquitoes in Africa, gene drive technologies could provide valuable new tools that are needed to eliminate the disease. Just as Africa played a seminal role in the development of innovative genetic approaches to insect control in the 20th century, it is poised to do the same in the 21st century. Continued testing and implementation of gene drive technology can establish Africa as a global leader in this field.